Assalamualaikum. Welcome to our channel Botany with YouTube. Today I am here, a PhD scholar. I am going to present a topic, Kingdom Monera. Firstly, I want to tell you about prokaryotes. The smallest and most common of cell are prokaryotes. You can observe here the structure of prokaryotes in diagram. Pili, capsule, outer membrane, peptidoglycan layer, plasma membrane, DNA and flagellum are the component of prokaryote cell. They exist almost everywhere on earth. They are cells that do not have nucleus. They do not have membrane-bound organelles. All prokaryotes are placed into one of the two kingdoms, Eubacteria or Archaebacteria. Botany with YouTube is our channel. If you are new in our channel, then please try to subscribe our channel. Firstly, Eubacteria. They make up the larger of the two prokaryotes kingdom. Generally, they are surrounded by cell wall composed of complex carbohydrates. Within the cell wall is the cell membrane that surrounds the cytoplasm. You can observe here the bacterial cell in diagram. Flagella, nucleus, cell wall, cell membrane, ribosome, cytoplasm, capsule are the component of bacterial cell. Cyanobacteria. They also known as blue-green algae. They are photosynthetic. They contain phycocyanin and chlorophyll A. They can be found nearly everywhere on the earth. They can survive in extremely hot environment and even extremely cold environment. Archaebacteria. They lack important carbohydrates found in cell walls. They have differentiate or different lipid in their cell membrane, types of ribosomes and gene sequence. Archaebacteria can live in extremely harsh environment. They do not require oxygen and can live in extremely salty environments as well as extremely hot environment. Bacterium shapes. They may be present in cocci, bacillus and spirillum. Cocci are spear-shaped bacteria, bacillus or rod-shaped bacteria, spirillum are spiral-shaped bacteria. Bacteria can be arranged in cluster, chain, colonies or pairs. Botany with YouTube is our channel. Share our channel, share our videos with your other friends, colleagues and researchers. Cell wall. To study bacterial cell wall, one must strain it. This is called gram staining. Chemical nature of cell wall can be determined by gram staining technique. Gram stain consists of two dye, crystal violet and the other one is saffronine. By finding out what color the cell produces when it is gram stained, you can figure out the type of carbohydrate in the cell wall. Movement. They move with the help of flagella. Flagella is tail-like structure that whips around the propel the bacteria. Cilia. Miniature flagella surround the cell that help to swim. and some type of bacteria show non-motile, and they are attached to the substratum or any surface. They don't show any type of movement. Bacteria and their energy. Autotroph. Some bacteria can make their own food from inorganic molecules. Some type of bacteria are heterotrophs. They consume organic molecules made by other organisms. Autotroph. Photoautotrophic. They are the autotroph trap the energy of the sunlight like Cyanobacteria. Chemotrophic. They are the autotrophs that obtain their energy from inorganic molecules. Heterotrophs. Chemotrophic. Heterotrophs obtain their energy by taking in organic molecules, then breaking them down and absorbing them. Phototrophic heterotrophs use the sun energy, but they also need organic compound for nutrition. For example, E. coli bacteria. Bacterial respiration. They may be obligate aerobes, obligate anaerobes, facultative anaerobes, cannot live without oxygen, must live without oxygen, can live with or without oxygen. Example of obligate anaerobes. An example of obligate anaerobe is some type of bacteria which produce toxins. If these bacteria find their way into a place that is free of air and filled with food material, they will grow very quickly. As they grow, they produce toxins, poisons, they cause toxicity. You can observe here in diagram the Botox. Botany with YouTube is our channel. Like our channel, like our video, and if you have any question, you can ask us in comment section, and you can share your point of view related to our topic in the comment section. Bacteria reproduction. In favorable condition, bacteria can grow and divide quickly. They can reproduce in the following ways: by binary fission process, by conjugation process, and spore formation process. Binary fission process in bacteria, you can observe here in diagram. Cellular organism copies its genetic information, then split into two identical daughter cells. Conjugation, you can observe here in diagram the process of conjugation. A type of bacteria sex. Two organisms swap genetic information that contain the information such as resistant to penicillin. Spore formation, endospore. The structure of endospore you can observe here in diagram. Spore coat, cortex, core wall, DNA, ribosomes. A type of dormant cell highly resistant to environmental stresses. Endospores are formed by cells in response to environmental signals that indicate a limited factor for growth, such as exhaustion of an essential nutrient. Importance of bacteria. Bacteria is often used in food, bread, cheese, yogurt. They also use in industry, for example in breakdown oil and some cosmetic procedures or industries. Symbiosis. Bacteria develop a close relationship with other organisms in which the bacteria and the other organism both get benefit from each other. For example, E. coli bacteria. This is found in human digestive tract. Other type of relationship, parasitism. Bacteria exploit the host cell, entering them. You can observe here in diagram. Mutualism. Relationship in which two species live together in such a way that neither harm. Bacteria in the environment. Nutrient flow. Bacteria recycle and decompose or break down dead material. Sewage decomposition. Bacteria capable of digesting the hydrocarbons in petroleum are often used to clean up oil spills. Nitrogen fixation. Process by which nitrogen in the atmosphere is converted into a form that can be used by living things. You can observe here how bacteria help in nitrogen fixation process. Thanks to visit our channel. Thanks for your concentration, for your attention. Good luck to all of you. Allah Hafiz.